y'all, it's Bella. So I wanted to show y'all what a typical day in the lab might look like for me. So I am a 4th year biology PhD student. So this is kind of specific to the STEM field. I'm sure everybody's lab is different. If you have a different PI, things may be much different. Your time schedules may be different. Of course, your research will be different. So I'm just showing you guys what my lab day looks like. So what I'm gonna be doing, I have to do an experiment. So I'm gonna be setting up an experiment. I do run my experiments for 24 hours. So I guess I'll just show you guys how I set it up, and then some little lab lead tasks, you know, like doing the centrifuge, going to the autoclave, even doing the dishes. So if you guys want to see that, let's keep watching. Okay, weird angle, I know. But for my research, I work with algae. So the first thing I do is I come into our little algae growing room. So my algae grows here in these flasks on this rotary shaker. So it gets constant movement, and then it gets UV. So it's on a 12-hour light day cycle. So our night is their day cycle. So when I come in, they're in their night cycle, and that's when they're most active. So that's when I work with it. So once I get the cultures that I want, I just make sure to turn the lights back off. So my work's gonna start in this laminar flow hood, and I have to talk to y'all before I turn it on because it's really loud. So it has a blower, it has lights, and it's going to sterilize the air that I'm working in so my algae doesn't get contaminated. And then we also add a flame in ours, so to further sterilize the air from any bacteria or anything that could contaminate my algae.
Ready or not, 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 here I come. Ready or not, ready or not, want me, ready or not, ready or not, here I come, ready or not, ready or not.
So I turned the blower off so y'all can hear me. So I'm gonna take those samples that I poured and I'm gonna put them in this centrifuge, and it's gonna spin them really fast, because what I want to do is separate my algae from the growth media that it was in and put it in experimental media. So it's gonna separate the algae from the liquid, and then I'm gonna pour it off, put it in experimental media. But I'm gonna show you guys all that, but I just wanted to explain like what I was doing. So I just have to put them in here, make sure that they're balanced. So you can see it speed up. So while that's running, I'm going to prepare the solutions that I have. So depending on what experiment I'm doing, I might make different chemical solutions to make different chemical gradients or whatever. But I do have to do a little calculation to figure out how much I need, and then I'm going to start working on that.
Ready or not, here I come, you can't hide. Gonna find you and take it slowly. Ready or not, here I come, you can't hide. Gonna find you and make you want me, want me, want me, want me, want me.
So the centrifuge is done. I'm going to unlock it and I'll show you guys what that has done. So you can see it separates my algae down there from the liquid, and I'm gonna pour it off.
Ready or not, here I come, you can't hide. Gonna find you and take it slowly. Ready or not, here I come, you can't hide. Gonna find you and make you want me. Ready or not, ready or not.
Of course I have to multitask here, so now on that second run I'm going to start actually setting up my experiment and the things that the algae are actually going to go in.
Here I come, ready or not, ready or not, want me, ready or not, ready or not, here I come, ready or not, ready or not, want me. You can't run away, and anywhere go, you can't run away, and anywhere go, you can't run away, all these stars I got obey.
So now that everything's in experimental media, I can start setting my experiment up. And one more thing I have to add, I can't share the details, like exactly what I'm doing with my experiments, because for one, it's my university's intellectual property. So when you're doing research, they own your research, they own my lab notebook that I'm about to go write in. So I just can't share too many details. Plus that would just be rude to me and my professor's hard work for everything we've come up with and that we're doing here and that I haven't published on yet. That would be very dangerous. So I can't share the specific details of my experiments.
Ready or not, here I come. You can't hide. Gonna find you and take it slow.
Okay, I'm done setting everything up. So I think it took me like an hour and a half, maybe kind of felt like 2 hours. This was my first time setting up 3 separate experiments. So now that we're getting close to me wanting to publish a paper, I'm trying to ramp up the work, ramp up the data, get as many results as I can, just trying to get it done. So now that I'm done setting up, I'm gonna clean up and then we'll go back to my desk. So now that I'm done setting up, I have to come to my desk and I'm going to write in my lab notebook every single little detail about the experiments I just set up so that I can remember any type of conditions or anything I need to put in a paper, or somebody can refer it back, or me and my professor can sit down and figure out what went wrong. That happens a lot. So I'm just gonna sit here and draw everything out. If I made new solutions, how much did I use, how many milligrams, how many milliliters, if it was water, if it was media, things like that. And I'm gonna get some extra data to go in here as well.
Ready or not, here I come. You can't hide. Gonna find you and make you want me. Ready or not, ready or not, here I come. Ready or not, ready or not.
So now that I'm done doing all my notes, so what I'm gonna do is make some new growth media and experimental media since I used it all this setup.
Ready or not, ready or not, here I come. Ready or not, ready or not. You can't run away. And anyway, so you can't run away. And anyway, so you can't run away. For these stars I got, oh baby, hey baby, 'cause I got a lot, oh yeah. And anyway, so my whole crew gonna know, oh baby.
Let me tell y'all, this is autoclave tape. So what it's gonna do, it has little white stripes on it. So when I put it in the autoclave and I take it out, these white stripes will turn black so that I know we got to the right pressure and the right temperature, so I know that my things are sterilized.
Hey baby, can't hide from love. Ready or not, here I come. You can't hide. Gonna find you and take it slowly. Ready or not, here I come. You can't hide. Gonna find you and make you want me. Ready or not, ready or not, here I come.
Okay, I hope y'all can hear me. So this is the autoclave, so I'm gonna load my stuff in. Since it hasn't been used like in the past few hours, it's cooled down, so I'm gonna have to wait for the temperature to get back up and for the pressure to get up. Next lovely task, I'm gonna gather up these dishes, all this glassware, and put them in the dishwasher. So my autoclave has like 30 more minutes, so I'm gonna sit here and I'm not about to do no work. I'm about to take maybe like a 30-minute break, probably sit on YouTube or Instagram. But this will be a great time, like if I had to fill out a form or something and get it submitted, or like return some keys or something little, like I could run in and go do it or something. But yeah, I'm about to get on Instagram or YouTube. What can I say? experiment has hit the 3-hour mark, I'm going to go back and check it, take some pictures, make some observations. I poured these agar plates, so they're already starting to solidify. You can see them kind of turning white, so that means they're getting hard. So while I'm waiting on those plates to dry and I'm waiting on the 6-hour observation point for my experiment, I'm gonna go get something to eat. So I could have brought my lunch so I didn't have to leave, or I could just go home. I could have went home and ate, cooked a little bit, chilled a little bit, so I have to come back at 5:45. So the dishes will be done at like 5:15. So when I come back, I'll take the dishes out, and I think that'll be the last thing I do in the lab. I have a little smoothie bowl here for lunch, almost dinner, but what I wanted to explain to y'all was that, so I'm going back for my 6-hour observation point, and then I'll go back for 12 hours, and then tomorrow will be my 24 hours. So every day does not look like this for me. Sometimes I'll go in for 10 minutes. Like tomorrow, I'll only be required to go in and take my 24-hour observation point. Or I could spend some time and discuss things with my professor. I could go in for 3 hours. I could be in there for 12 hours. I could go at 2 a.m. Like I said, today is Sunday, so I'm doing work on a Sunday, Saturday, which I prefer to work on the weekends because I can park closer and ain't nobody else in the building. I don't have to wait on the autoclave. I don't have to wait on the dishwasher, none of that. But I was trying to decide if I was gonna go back home or go to the lab, and I think I'm gonna just go to the lab because all I have to do is read a paper and like study up on a paper, probably start putting these pictures together. So I could do that anywhere, but since I gotta go back, I will be home for like an hour, so there's really no point in driving home.
We got beef on the trap.
Work. To me, I be work. You see me do me dirt. So me better work.
All right, so I got the dishes. I got my 6-hour time point. What is that clicking noise? Anyway, so all I have to do is put the dishes up in the cabinet, and I'm gonna head home, do some laundry, do some of my dishes at home. I'm gonna probably chill because this was a pretty long day for me, a lot of work. I'm gonna end the video here. I hope you guys liked it and enjoyed it. If you did, if you enjoyed this type of content from me, go ahead and give it a thumbs up. Don't forget to subscribe, and if you guys have any questions that I can answer, I would be happy to answer. I'll see y'all next time. Bye. I'm gonna put the dishes up.
When you walk a line, I'm in the cafe, drive me, I deserve you. No time to have you lurking. You act like me, you don't like it. You know I dealt with you the nicest. Nobody touched me in the righteous. Nobody text me in a crisis. I believed all of your dreams are duration. You took my heart, all my keys, and my patience. You took my heart, I must leave a decoration. You mistaken my love, I brought for you for foundation. All that I want